Tonight, we explore the remarkable story behind one of medicine's greatest breakthroughs, the quiet, accidental discovery of penicillin, and how a chance encounter with a simple mold transformed the fight against deadly infections forever. Welcome to Slumber Scapes, where the wonders of medicine unfold in the quiet of night. Settle in and join me on a gentle journey through its marvels. And if this calm exploration feels like home, stay with me for more soothing and peaceful stories ahead. Close your eyes and imagine a world where even the smallest cut could end a life. The year is 1920. The air smells faintly of coal smoke in the crowded streets, and in the quiet corners of hospitals, the sound of muffled footsteps mixes with the low hum of whispers. People speak in cautious tones about illnesses with no cure: pneumonia, blood poisoning, tuberculosis. You are standing in a ward warmed by glowing lamps, blankets pulled tight over patients still weak from fever. Doctors in stiff white coats pass from bed to bed, but their tools are few and their medicines limited. The scent of antiseptic is sharp, masking but never erasing the smell of infection. Here, survival is uncertain. You feel the weight of those times. How parents feared for their children's fevers. How a soldier's wound often meant more danger from infection than from the battlefield itself. In this world, medicine is a mixture of careful nursing, experimental remedies, and hope. There is no penicillin yet, and so each recovery feels like a small miracle, and every life lost a reminder of infections that the body alone could not defeat. This is the medical stage upon which our story begins, a time just waiting for an unexpected hero. Now let's turn our gaze to a quiet laboratory in St. Mary's Hospital, London. A tall man with gentle, steady eyes leans over a workbench scattered with glass plates and flasks. This is Alexander Fleming, a bacteriologist with a tendency to notice what others overlook. His friends would tell you of his soft-spoken nature, his dry wit, and his meticulous hands that could coax secrets from the smallest organisms. You hear the faint scratch of his pencil on paper, the rhythmic tap of glass pipettes against the bench, and the comforting tick of the wall clock. Nearby, colonies of bacteria grow in neat rows upon petri dishes, his silent companions in a lifelong study of microbes. Fleming had already served in World War I, treating soldiers whose wounds were often beyond surgical help. That memory stayed with him, the helplessness of watching infection spread when nothing more could be done. He carried that memory quietly, like a weight in his coat pocket. He was not searching for a wonder drug on that warm September morning in 1928, but life sometimes whispers the extraordinary into the ears of those who are simply paying attention. Picture a lab lit by early autumn light. The air smells faintly of dust and agar. Fleming returns from a 2-week holiday to find his desk cluttered with stacks of labeled petri dishes, an untidy result of his own hasty departure. As he moves through them, his practiced gaze spots something unusual. One plate, left uncovered for a time, has grown a delicate bloom of greenish-gray mold at its center. But that is not what arrests his attention. What catches his eye is the clear halo surrounding the mold, a perfect, empty space where bacteria have failed to grow. Fleming tilts the dish toward the light, tracing that translucent ring with his eyes. It is as if the mold has cast a protective circle, silently pushing the bacteria away. The detail is small, easily dismissed, but to him it sings with quiet mystery. No fanfare, no lightning strike. Only a man, a desk, and a coincidence. And yet, this scene holds the first page of a new chapter in medical history. In the days that follow, Fleming studies the petri dish with patient curiosity. You might picture him seated under the soft glow of a desk lamp, the room cool in the early morning, his breath faint in the stillness. He transfers bits of the mold to fresh plates, watching how it grows. Under the microscope, the bacteria are numerous and eager until they near the zone of the mold, where they stop as if halted by an invisible barrier. This mold, he identifies, belongs to the Penicillium genus. He begins calling its mysterious substance mold juice, a humble name for what will become the world's first antibiotic. The term antibiotic itself was not yet part of everyday language in these early days. It was simply an oddity worth exploring. Still, the question lingers like the faint, musty scent of the mold itself. What is in this green bloom that can so effortlessly hold back the tide of infection? One afternoon, while arranging his notes, Fleming gives it a name. From Penicillium, the mold, he draws the word penicillin. The syllables feel clear and purposeful, carrying the sturdiness of its scientific roots and the simplicity of its appearance. By now, the mold's scent is familiar, earthy, almost like a damp forest floor after rain. It clings faintly to the air in his workspace. Each experiment reinforces the same sight, a neat perimeter where bacteria dare not cross. He writes about these findings, carefully describing the mold's bacteria-killing ability, and notes that it seems harmless to human cells. It's a thought that feels like a soft light in a dim room, the possibility that something so ordinary as a mold could be a healer. Though the world does not yet realize it, the naming of penicillin plants a seed. In quiet tones and handwritten pages, the greatest medical revolution of the century begins to take shape. In the days that followed his initial discovery, Fleming's lab became a place of patient repetition and careful observation. The scent of agar mingled with the faint, earthy aroma of the mold as he shifted between his workbench and the dim glow of the microscope. You imagine the cool, polished surface of a petri dish beneath his fingertips as he transfers tiny flecks of penicillium to fresh cultures, each one an experiment in miniature. He dilutes the extract, his mold juice, and watches as bacteria either thrive or falter in its presence. Yet the substance is fragile, breaking down quickly, losing its power outside the cozy home of the mold. Its instability frustrates him, like a candle that gutters out before the darkness is pushed away. He begins to see its potential, harmless to human cells, yet merciless to certain bacteria. But the path from an intriguing observation to a usable medicine is uncertain, slippery, and prone to vanishing into nothing. For now, the mold juice is a whisper of a cure the world doesn't yet know it needs. When Fleming publishes his findings in 1929, the scientific paper is thorough, the evidence compelling, but the world's response feels muted. The article sits quietly in the pages of a medical journal, its significance overlooked like a rare gem lying unnoticed among stones. You picture the London medical scene of the time. Lecture halls with rows of polished wooden benches, the faint scent of chalk dust, the rustle of paper as notes are taken. Discussions orbit around more immediate research, more fashionable pursuits. Fleming speaks at meetings, describing the strange mold and its bacterial halo, but most listen politely, then return to their own work. Some dismiss it as a laboratory curiosity, interesting but impractical. In those moments, the discovery comes dangerously close to fading into obscurity. The mold waits in glass containers at the back of a shelf, carrying a gift for humanity that, for now, few are ready to receive. One of the great challenges lies in coaxing penicillin into a pure, stable form. The mold grows reluctantly, producing only small amounts of its potent extract, and the active ingredient proves delicate, degrading with heat, light, or even the wrong handling. In the cool chalk-smelling lab, Fleming and fellow researchers work with glass funnels and beakers, measuring droplets in trembling increments. They try to separate the life-saving essence from the rest of the mold's broth, but each attempt leaves them with too little to test on patients. The process feels a little like trying to capture the scent of a rose in a jar, the key element always threatening to vanish. Eventually, Fleming shifts his focus, believing the work, while important, might require techniques beyond his reach. For now, penicillin remains an elusive miracle, its potential preserved in notes and fading cultures, waiting for fresh eyes and new hands to pick up the trail. As the 1930s unfold, penicillin rests in the background of medical research, like a photograph tucked away in a drawer. The Great Depression presses in, resources are scarce, and the medical community's attention drifts to other challenges and discoveries. You can almost hear the slow tick of the clock in the quiet corridors of St. Mary's. The mold cultures sitting unused on a shelf, their green surfaces fading to pale. The story could easily have ended here. the chance event of 1928 remembered only as one of Fleming's many small contributions. But history has a way of circling back to what matters. All it would take was the right combination of urgency, technology, and determined minds to breathe life back into the dormant discovery. That moment was coming, and it would arrive from a place and a time where the need for such a miracle was impossible to ignore. Long before penicillin reached its golden era, the experience of World War I had planted a seed in the minds of soldiers and doctors alike. Fleming, like many medical officers, had seen firsthand how infection could turn a survivable wound into a fatal one. You imagine the makeshift hospital tents near the front lines, the scent of damp canvas, the constant murmur of voices, the sharp tang of carbolic antiseptic in the air. Surgeons worked tirelessly, but without effective antibiotics, their knives could not outpace infection. Those memories lingered in the post-war years, shaping an unspoken promise among many in the medical field. If ever there were a way to halt infection safely, it must be found and shared. By the time the world edged toward another global conflict, the urgency returned in full force. War would once again create wounds that cried out for more than bandages and hope. And somewhere, in the green bloom of an old petri dish, the answer had already been waiting. Through the early 1930s, Fleming continued his work at St. Mary's with steady diligence. You can picture his modest laboratory, glass jars lining the walls, the faint smell of disinfectant in the air, and on one corner shelf, petri dishes of penicillium, quietly holding their secret. He spoke of penicillin occasionally, but no new breakthroughs came. The purification problem remained unsolved, and without that, large-scale studies or treatments were impossible. More pressing day-to-day research tasks drew his attention away. Outside the lab, life moved on. Seasons changed. The Thames glinted in summer sun, then lay muted on the winter fog. In these years, penicillin was a ghost in Fleming's career, present, remembered, but silent. Even so, careful records stayed tucked within his files. Perhaps somewhere in the back of his mind, he held on to a soft intuition that this strange mold-esque story wasn't over yet. In 1938, in the city of Oxford, two scientists, Howard Florey and Ernst Boris Chain, encountered Fleming's paper from a decade earlier. Under the high-arched ceilings of the Dunn School of Pathology, the room smelled faintly of waxed floors and the metallic Thames of lab instruments. Chain, a biochemist with restless curiosity, saw in the old study the spark of something transformative. Florey, methodical and quietly ambitious, agreed it was worth pursuing. They gathered a small but skilled team, including Norman Heatley, a young researcher quick with his hands and inventive with equipment. Together, they decided to revisit Fleming's mold juice, not as a curiosity but as a goal, to purify and harness penicillin into a usable drug. With the hum of incubators in the background, they began the painstaking work, unaware they were standing at the threshold of a medical revolution. By the time their early results proved promising, World War II had begun. Outside the lab, the world shook with uncertainty. Inside, a different battle raged, not against an enemy across the sea, but against scarcity and technical limits. Penicillin's yield was punishingly low. Gallons of nutrient broth produced only tiny, precious droplets of the active substance. The scientists improvised, using bedpans, milk churns, and even ceramic vessels to culture the mold in larger quantities. The lab air grew warm and moist, filled with the earthy aroma of growing penicillium. Days blurred into nights as extractions were performed and failures logged. Still, each drop was worth the effort. Soldiers at the front would soon need what these lab benches could produce. They were not just racing time, they were racing death. The first human trials in 1941 were both hopeful and heartbreaking. You picture a dimly lit hospital ward, the soft shuffle of nurses' shoes, and the quiet groans of patients fighting severe infections. A policeman named Albert Alexander, suffering from a deep infection after a minor injury, received the first doses. The change was remarkable. His fever eased, his strength returned. But the supply was so small that treatment ended too soon, and the infection returned, claiming his life. Even in loss, the message was clear. When penicillin was there, in enough quantity, it could turn the tide. Soon after, soldiers with infected wounds on the battlefield were given the drug, and many walked away from injuries that just a few years earlier would have been fatal. In those moments, penicillin stepped from theory into humanity's hands. By 1943, collaborative efforts between British and American laboratories brought penicillin into wider production. Imagine walking into a wartime hospital now. The antiseptic scent still lingers. The sound of footsteps echoes, but on metal trays, beside the bandages and syringes, are small vials of golden liquid. Nurses prepare careful doses. Doctors speak in hushed, hopeful tones. Patients who once faced the knife for amputations could now heal intact. Mothers recovered from serious postnatal infections. Children survived pneumonia that once seemed unstoppable. The war raged on outside, but within those wards, penicillin was quietly rewriting fate. A healing bloom carried from a forgotten petri dish into the heart of medicine. Step into a wartime surgical theater in the mid-1940s. The lighting is warm but low, casting soft pools above the operating table. You can sense the quiet focus in the room, the muted clink of steel instruments, the smell of sterilized gauze, the faint hum of a ceiling fan. Surgeons now operate with a quiet confidence unknown just a few years earlier. Before penicillin, operating on a severely injured limb often meant racing to stay ahead of infection. Amputation was common. Now, careful cleaning, stitching, and a measured injection of penicillin could turn a dangerous wound into a healing one. The presence of penicillin doesn't erase all risk, but it changes the odds. The recovery wards are fuller than before, fuller with the sounds of breathing, conversation, and the rustle of sheets rather than the silence of loss. The operating theater is no longer just a place of urgent rescue. It becomes a space for repair and restoration, edged with new hope. As wartime successes spread through military channels, news eventually reaches the public. Newspapers begin to print hopeful headlines. New drug fights, deadly infections. You can imagine people in small British towns or bustling American cities reading those words over morning tea or coffee, hope tinged with disbelief. After all, for centuries, infection was as old as humanity itself. The thought that it might be tamed sounds almost like fiction. In community clinics, doctors begin to prescribe penicillin for pneumonia, syphilis, and life-threatening sepsis. Results are swift, often dramatic. Families visit loved ones expecting the worst, and instead walk home together. These stories ripple through communities, and though supplies remain limited at first, the vision of a world where common infections are no longer feared begins to take root in the collective mind. A new era. The antibiotic age is being born. As production scales up after the war, penicillin reaches civilian hospitals across continents. You can picture crates marked for shipment crossing harbors, the salt air mingling with the faint medicinal scent of their precious cargo. In tropical climates, it begins saving lives from bacterial diseases common in warmer regions. In colder countries, it turns winter pneumonia from a dreaded killer into something treatable. Infant mortality rates drop. Post-surgical recovery rates soar. What was once a single petri dish in a London lab now spans the globe. Each vial a quiet answer to human vulnerability. The numbers are staggering, but the reality is even more profound. Behind every statistic lies a person, a family, and a story rewritten by a simple mold's secret gift. By 1945, the world can no longer overlook the scale of this transformation. The Nobel Prize in Physiology or Medicine is awarded jointly to Alexander Fleming, Howard Florey, and Ernst Boris Chain. The ceremony is dignified and calm, held in Stockholm's winter light. You imagine Fleming standing modestly in his dark suit, accepting the honor with quiet gratitude. In his speech, he warns, in gentle but prescient words, that misusing this drug could one day lead to resistant bacteria. The applause is warm, but the message is deeper than celebration. Penicillin's triumph comes with a responsibility to use it wisely. Around the world, laboratories are already seeking the next miracle, inspired by what this trio has achieved. Now that penicillin is mass-produced, pharmaceutical companies refine it into new forms: powders for reconstitution, tablets, and injectable solutions that can be stored and transported far from the lab bench. You can picture a pharmacist in a white coat, carefully weighing doses, the scent of paper envelopes and antiseptic hanging in the air. Penicillin, once fragile and fleeting, is now sturdy enough to travel in a doctor's bag to a rural village, or in a soldier's kit to a battlefield. The journey from mold juice to reliable medicine is complete, yet still evolving. Chemists experiment with modifications, exploring whether other compounds might work in similar ways, paving the road for dozens of new A revolution in medicine has begun, and it shows no signs of stopping. In the years after its introduction, penicillin didn't just save lives, it changed the way scientists thought about medicine itself. Picture research labs in the late 1940s, sterile benches bathed in a steady glow, the soft worry of early centrifuges, glassware clinking gently under careful hands. Inspired by the mold that halted bacteria, entire teams began scouring the natural world for other microbes or fungi with similar secrets. Forests, soil, even ocean mud became fields of exploration. Every sample was a potential key. Every petri dish a blank page waiting for nature to write a new cure. Penicillin had shown that nature's smallest inhabitants could wield immense power, and that with patience, observation, and cooperation, humans could learn to use that power for healing. It was as if the door to a new era of possibility had been swung wide open. By the early 1950s, more antibiotics arrived: streptomycin for tuberculosis, tetracycline for a range of infections, chloramphenicol for typhoid fever. In each case, the lessons learned from penicillin's discovery and production shaped faster, more efficient development. The air in these expanding pharmaceutical workshops carried the warm scent of sterilizing steam and the hum of constant activity. Shelves filled not just with penicillin but with new bottles and vials, each promising to conquer another once-feared disease. Hospitals began to feel like fortresses against infection. Doctors could, for the first time, choose from a growing arsenal rather than relying on a single precious drug. The age-old balance between human vulnerability and microbial threat had shifted. Even so, the story was far from over. Nature's tiniest adversaries have always been quick to adapt. Imagine you are small enough to travel inside your own bloodstream, drifting gently like a leaf on a slow river. Around you, red blood cells float past in rhythmic waves. When you take penicillin, its molecules journey through this river, seeking out the bacteria causing harm. For certain bacteria, penicillin acts like an unseen craftsman dismantling the walls of their microscopic homes. Without those sturdy walls, the bacteria weaken and dissolved, their threat fading away. What makes penicillin remarkable is its precision. It can harm those bacterial walls without touching your own cells, leaving your body's tissues unharmed. In this way, the drug works hand in hand with your immune system, while penicillin removes the invader's defenses. Your own white blood cells move in to finish the job, quietly repairing and restoring balance within. Healing is more than just the removal of illness. It's the body finding its quiet equilibrium again. Picture lying in bed, the air warm under the blankets, your breathing slow and deep. Beneath that stillness, the penicillin you took hours ago is still moving within you, each molecule helping to hold the line as your immune system rebuilds strength. Your body's own repair crews, cells that seal wounds, clear debris, and regenerate tissue, work in steady harmony. Penicillin does not replace these rhythms, and supports them, giving your natural defenses the space and time to restore health. It's a partnership as old as life itself, the body striving for wellness, and now, thanks to science, a companion to help it along the way. In his Nobel speech, Fleming's voice was measured but firm. Overuse or misuse of penicillin could teach bacteria how to resist it. This warning, spoken in the hopeful aftermath of war, seemed almost hypothetical, but in time, it proved prophetic. In hospitals, laboratories began to see certain bacteria no longer halted by penicillin's barriers. These resistant strains spread quietly at first, a subtle undercurrent in the tide of medical triumph. The air in those labs was tinged with apprehension. Petri dishes once lined with clear bacterial halos now showed colonies pressing against the mold's territory. Fleming's words echoed. The miracle of antibiotics could be undone if humanity forgot that microbes also learn, adapt, and endure. It was a reminder that every gift, even one as great as penicillin, must be cared for wisely. Today, in hospitals buzzing with soft machine beeps and the quiet rhythm of breathing monitors, a new challenge hums beneath the surface. Bacteria that can withstand many antibiotics, including penicillin. These superbugs are the result of decades of adaptation. Nature's reminder that even the smallest life forms are endlessly resourceful. You can imagine a lab technician carefully sealing samples, the sterile air tinged with the faint scent of ethanol. Under the microscope, these resistant bacteria look no different from their weaker cousins. Yet they stand firm against treatments that once ended infections swiftly. Around the world, scientists share data, search for new drugs, and revisit old ones, using the collaborative spirit that once brought penicillin to life. The fight is ongoing, and hope still threads through each experiment, each trial, each idea that may turn the tide once again. Walk into a modern clinic and you'll see penicillin still there, perhaps in a vial on a refrigerated shelf, perhaps written on a prescription slip in neat blue ink. Its presence feels almost ordinary now, but the truth is that without it, much of modern medicine would crumble. Surgical procedures, organ transplants, even routine dental work rely on antibiotics to keep infection at bay. Vaccination campaigns and public health strategies work alongside these medicines to protect communities. Every life saved by other antibiotics still owes something to penicillin's story. It was the proof of concept, the first clear sign that we could stand beside nature, not only in observation but in partnership, to heal. With every prescription comes a choice: to use antibiotics only when truly needed, and to complete the course so that bacteria do not get the chance to grow stronger. In the quiet of a doctor's office, you can picture a caring discussion, a physician explaining to a patient why an antibiotic won't help a viral cold, or why finishing all the doses matters. The advice may sound simple, but it is part of a vast, collective defense. Just as Fleming's warning echoed across decades, so too does this modern principle, that medicines are not infinite gifts, they are shared resources, sustained by the choices we make together. Scientists now explore paths that move alongside or even beyond antibiotics, treatments that use bacteriophages, specialized enzymes, or immune-boosting therapies to target harmful microbes. In one research center, sunlight filters through high windows onto rows of incubators, each holding possibilities for tomorrow. The air vibrates with quiet determination, much like the Oxford lab decades ago when penicillin itself was a fragile dream. The next breakthrough could come from a remote forest soil sample, a deep-sea organism, or a genetic innovation. The future of infection control will likely be a tapestry woven from many kinds of treatments, each carrying the legacy of that first mold-streaked plate. And so, we return to that autumn day in 1928, to a cluttered desk and a forgotten petri dish. From that small and quiet moment unfolded a chain of events that changed the fate of millions. Penicillin became more than a medicine. It became a symbol of what can happen when curiosity meets patience, when chance meets preparation, when knowledge is shared in the spirit of care. Tonight, as you rest, remember that every gentle breath you take is part of this same story, a world where lives are longer, safer, and closer, because a man noticed a halo of empty space around a patch of mold. May this reminder bring a calm gratitude, and may you drift into dreams warmed by the quiet truth that discoveries, even the smallest ones, can change the world.